Good afternoon, everyone, and thank you for attending our Agribiome Seminar. For those who don't know me, my name is Samat Ahmed. I'm a new faculty in Department of Microbiological Science. And our first Agribiome Seminar, Dr. Tim McAllister introduced us the concept of the cattle microbiome and how the cattle microbiome could be used to mitigate some of the challenges that we are facing, such as antimicrobial resistance and the environmental impact of the cattle production. So now, today we're gonna get into a little bit deeper on the gut microbiome of cattle. And for this topic, and I'm humbled to bring Dr. LeLuo Guan. Dr. Guan is joining us from Edmonton, Alberta, Canada. She's a professor and associate dean at the College of Agricultural Life and Environmental Science. And I assume some of you already got a chance to read her short bio from our seminar announcement. So I will be very briefly outline some of her achievements. And Dr. Guan is one of the leading scientists in cattle research, and her research program is unique, I think, in a way that focuses on both host genome and microbiome, and using cutting-edge techniques, including multiomics, to understand how the host microbiome interact and their role in defining cattle performance and production. So Dr. Guan has published more than 200 papers, and some of her papers are published by highly respected journals, including ISME and Genome Biology. She has trained more than 50 graduate and undergraduate graduate students and postdocs, and luckily recently I have become one of Dr. Guan's mentee, and she's serving as an external mentor for my NHI COPRA project. So today, Dr. Guan's topic is the role of gut microbiome in cattle health and production, whether microbiome is driver or passenger. I like this title, and I personally believe that the gut microbiome now can have the driver's seat from host genome. And yes, we made a significant progress in improving animal health and production through genetic selections, but I believe that harnessing the rumen microbiome may also able to improve cattle production and health. Now let's hear from Dr. Guan, and Dr. Guan, floor is yours. And if you have any questions during the talk, you can type it on the chat box, and we'll have a chance at the end of the seminar to answer those questions. Thank you. Thanks, Dr. Guan.
Thank you, Samat. Thank you for Dr. Ahmad's very kind introduction and also invitation, and to give me an opportunity to share some of the research we're doing at University of Alberta. I didn't know that team has talked about cattle a little bit already, so I apologize ahead if some of my contents could be a little bit overlap. But also, I've been thinking how to tackle this topic in the past. So to cover everything, I thought is very challenging. So I actually prepared 2 sets of slides, 2 actually slides topics. So I'll try my best to do. It might be challenging to do both of them, but I hope there are some concept or overlap. But I'll start with the first one, and then we'll see how it goes, and then we can move on to the second one. So I think the title, as Dr. Matt mentioned, I'm trying to talking about with all this extensive study have been reported that microbiome play a very important role in animal production and health. And, can we actually make a conclusive conclusion that the microbes actually drive those traits? And can we actually manipulate those microbes to improve the productive and health-related traits. So apologize, that's what I said, if this is overlapping. So I actually, the first slide is more like the story from the rumen. I like to share all the past kind of experience, what we had, and hope that can give you some ideas and or insights to include some of the perspective into your coming research. So I'll start with some introduction. I think if Dr. McAllister has already introduced, I think for many of us, especially even for my own students, there is always a struggling to understand the terminology about microbiota and the microbiome. And also I find out when we even write a manuscript, sometimes we submit manuscript and we use microbiome, the reviewer says, no, it's microbiota. So I think now is the time now we need to actually clearly identify or differentiate those two terminology. So the microbiota, well, likely refer to the composition of a microbial community, and a microbiome in general, now we refer them as a total genetic elements like a genome or the genes of a particular microbial community. So if we think about the rumen, I know that if this is maybe a little bit overlapping. So when we're talking about rumen microbiota, we're usually talking about the composition. I'm not for sure. Can you see the arrow when I move? Okay. Great, thank you. So there are bacteria, and there are archaea, there are protozoa, there are fungi, there are bacteriophage. Okay, so when you see here, many of the past study are mainly focused on bacteria, archaea, protozoa, and the role of fungi in the rumen is still very limited. And together, recently in the human relay study, there was an increasing interest in looking at virome. As part of a virome, bacteriophage could also play a very important role in driving the overall microbiota composition. And for rumen microbiome, usually we refer them as microbial genomes and genes within the microbiota. Okay, so when we do microbiome research, I think it's very important for us to plan and prepare very carefully, because that there are so many variables or steps can affect outcomes of the microbiome research. So I like to include some of a background or introduction on how we should or tackle when we try to plan to study microbiome, regardless as a rumen or a gut. So when we think about microbiome, there are different populations. I think if I know team, Dr. McAllister may also mention about this. Within the microbial community, there are different populations. So using rumen as example, their population actually associated with different ecological niche. For example, there is a large population attached to the feed. There are certain populations, they are just free floating. And then some populations, they actually attach to the rumen epithelium. So this is actually similar to all gut microbiome or gut microbiota. Even in the lower gut, there is a digesta associated, there is a free floating, there is also mucosal attached microbiota. And they actually can be significant different in terms of their composition as well as their function. So this is one of the essential questions when you think about microbiota, which population are you looking at? And which population you think that it may play a role in driving or changing some of the function of the microbiome. And in addition, how those different populations respond to the diet or environment or treatment differently, that could also impact overall function of a particular microbiome, here, rumen microbiome. So in addition to those essential population ecology, sampling is very important. This is what we do. This is actually all my student. Apologize, I was the one taking pictures. But I do go sampling all the time, especially for every first sampling during the year, I do go to make sure everything is collected properly. So you can see here, this is the whole gut. We actually collect samples in a gut room in a slaughterhouse. And you can see this is the whole gut of a beef cattle. And this is the rumen. This is huge. It is just gigantic, is huge. I always tell my student, you know, if I can climb into the rumen, I can feed there really well because I'm very small. So you can see here. So that's why when you think about doing microbiome research, right? This is huge. Where we should collect samples, and you know how many samples we need to collect, and they can be representative, and also you know how often we need to collect samples. So there are lots of questions we need to think before actually we start to plan or collect samples for microbiome research. You might wondering why, because the sampling time point will significantly affect what you try to address. So for example, if you look at, this is just showing a cattle, the rumen, fracturation rumen environment. If cattle are fed with a high forage diet, the pH is relatively stable. So when the pH is stable, likely that rumen environment has less impact on the microbial composition shift. But when you look at cattle are fed high grain diet, the pH change very dynamically through that 24-hour feeding cycle. So this pH can significantly affect the microbial composition. So think about if you collect the rumen samples here before feeding versus you collect samples 3 hours after feeding or 6 hours after feeding, your rumen microbial composition may shift significantly to respond to that rumen environment change. So indeed, you can see here, people have reported that different population so can be detected through that feeding course. So some of you may wonder, so let's say this is the microbial community within the rumen, within the same animal, where their composition actually changed that much, okay? So richness means the number of species may not change that significantly through that feeding cycle. However, the number, so the evenness or the abundance of that membership may change in response to the feeding cycle. However, when we're trying to do microbiota research, we always have to question, you know, how the nutritional management change microbiota. We actually compare which species is high or low. So this is actually really critical for us to think that what we want to answer is that we answer that it respond to the feeding or is respond to particular rumen environment. So I think how to determine when to collect a sample is actually really critical. So for us, in the past, we actually collect multiple time point in the nutritional feeding trials. Sometimes we test whether there is a significant difference, and then we combine them together. Second question, same for the, well, this is for rumen, but also for the gut as well, the lower gut. We know that lower gut is huge, could be long as a couple of meters long. So which section should be the representative? So when you need to think about the rumen, there is all sorts of different sections. And then here you can see even the bottom, there is the fiber mass, there's a gas, there's a liquid. So where should we collect samples to study rumen microbiome? So this is actually another critical point we need to pay attention to before we perform any research. So at moment, we actually, if we use cannulated cows, we in general trying to collect samples at about 5 locations. So the ventral sac, cranial sac, I think is usually 1, 2, 3, top, bottom, which one is front? Sorry, front, back, and the middle. We try to collect 5 locations. Same for when we slaughter animals. But if we use tubing, that's always sometimes a challenge. We actually get criticized when we're using the tubing through the esophagus, whether the tubes reach the same location, we actually can collect rumen samples to be representative. So you can see here clearly that depends on where you look for the rumen microbiota. You actually get samples that are associated with different proportions. Like here is more associated with fluid, here is more associated with solid. So you can see here that microbiota can differ significantly among different fractions. Okay, so in addition to that, I think this question has brought a lot of concern recently as well, because as I said, we want to determine a conclusive conclusion that microbes are a determining factor. But because we're doing sequencing, so most of the results are based on the DNA extraction and the sequencing. And as many of us working as a livestock, dealing with animals, we cannot really collect samples at the same time. We have to collect samples at different time, and we actually process samples may also different times. So you also need to be aware your sample storage method and the processing can also affect your sequencing outcomes. I know this is a very small paper, but if you look at this paper, it clearly indicate if the samples stored at -20, -80, or through the freeze dry, and you use the same method to extract DNA and you do sequencing, their microbial profiles can somehow different. So what that means, so you see that when I try to introduce those variables, make things very complicated, is what I want to say is when we do microbiota research, we really need to take into account many factors can affect the microbiota determination outcomes. So if this is the case, I would like to suggest that when you do statistical analysis, if your samples were collected at different time, stored on the different time point, and it's very important for you to include a batch effect. So you can actually test whether there is a batch effect that could affect your data. And then if there's no batch effect, then you can be more convinced that likely your microbial analysis is accurate, or there is no bias due to different batches doing extraction, different personnel to analyze, to extract DNA and processing the samples. Okay, I'll just wrap up in a few slides talking about the very commonly used method right now. Everyone seems like doing it, the amplicon sequencing based method. So we all use this because now it's really quick and it's also very costly effective, but also you really need to be aware there are some limitations of the method and also how you interpret your data. So here, make sure that the primers that target bacteria and archaea for ruminant research that actually are significant different from human, many other monogastric animal research. I didn't include a slide, but actually if you see the papers, there are many people use that V3 region, the variable region of the 16S RNA gene, to profile rumen microbiome actually is largely biased because that primer actually cannot separate bacteria and archaea. And also, it does not really provide a better classification of rumen microbiota. I think one of the researchers at UPenn, and she actually also published a paper a couple of years ago compared to different primer sets. And she found out, I think it's Dr. Pita, and she found out that actually the primers target V1, V4 region, which is designed by Agri Research New Zealand, which has been used for global rumen census that actually represent the most comprehensive microbial profiling for the rumen bacteria. And also, now go back to my first slide. Indeed, the states for all the 16S RNA gene-based research, we actually refer it as meta-taxonomic analysis. So they were not metagenome, they were not actually really microbiome, they were more related to taxonomic analysis. So we refer it as meta-taxonomic analysis. So again, because we're talking about the, we want to identify the microbiome's driving factors, so it's really critical for us to identify microbiota at a proper taxonomic level. But also I want to bring this up because when you're trying to submit your samples for sequencing, I strongly suggest you include a negative control for all the future sequencing analysis, because that even the DNA extraction kit can introduce contaminants, and even the sequencing, they actually has carryover contamination effect. And also potentially the PCR bias depends on the primers and your sample nature. So it's very important to include a negative control. And I think most recently, many researchers also include a kind of a MOC community as a positive control to evaluate the impact of the sequencing could affect the 16S RNA gene based microbial compositional analysis. And another important thing is when we study microbiota or microbiome is important, we use proper database. I guess as one of the editors for FAM's microbial ecology, for my responsibility in terms of animal microbiome and also my role in Animal Microbiome Journal, it has been brought up that for animal research, the human database can introduce lots of biased information. So using rumen as example, if you do not use a rumen specific archaea database, when you're trying to look at rumen archaea, you actually generate a lot of information about taxa they don't live in a rumen at all. So this is actually really important when you do the microbiome research. It will be very important if you can actually develop an in-house enriched database for your species like pigs or chicken potentially. We actually use a very rumen specific database we actually created for our metagenome analysis to really generate information that is rumen specific and targeted. Okay, so another thing is I like to add that for to publish the high impact journal that 16S based data are now considered just a descriptive. So you need to have validation either through the qPCR or culture-based or functional aspect to together with your microbial composition analysis, and that could be considered to be published at high impact journals. Sorry, I used a little bit long time for that. So I hope just give you some ideas how critical when we study microbiome, we are not just collecting samples, extract DNA, especially I know that many researchers in certain area, they just even send samples to some company, they get DNA, they get sequenced, and then they submit a paper and they use the data completely done just by company. So I think it's really critical for all of us to think how those data, how we can prepared for to study microbiome. So then I'll move on the story about talking about the role of rumen microbiome in cattle feed efficiency. So when I start to work on this, and actually this is the first paper I published in 2008, and actually it took me 3 years to publish this paper. This is a very small paper. And I also remember when I submitted this paper, I got 5 reviewers. And this is, you can see, it's a research letter. And at the time, I got feedback from 5 reviewers, and I wrote replies about, if I remember properly, that's almost close to 30 pages of replies, because at the time it was, I was criticized that rumen microbiology is microbiology, feed efficiency is a feed efficiency traits, and somehow, it seems like I just, trying to put them together, it's kind of a bizarre idea, or they may not really directly contribute and so on. But anyway, so this is actually took 3 years. I submitted the first draft in 2005 and finally published in 2008. But I thought that was actually a really good experience I like share with the students. And sometimes I know that we all need papers or manuscript to publish, but sometimes I think is if you can strengthen the manuscript when it's rejected, and if you can develop a better idea or story, and eventually it can be published and also can be recognized your novel idea and a contribution. But anyway, so since then, what we have done? Okay, so because we talk about rumen microbiota, their important role, and I think you guys may pay a little bit attention, I'd add a 2 cycle in the right bottom. So I'm not going to explain the others. I want you think about what does this core and pan microbiota mean. Okay, so this is today's focus on, when we think about the microbiota, that there are microbes exist in all the individual through the population. This is the same, actually, we also found for the lower gut microbiota. And also there are microorganisms only, exist or detected in particular individuals. But how can we identify who may contribute to the function, and do they all contribute to the phenotype, or do they contribute phenotype at different level, and which one is more important? And if we want to manipulate them, should we manipulate those core, or should we manipulate the pan? Because this actually gives us very different approach to manipulate. Okay. So I'll just move on to the feed efficiency we have been studying. So we actually, in the past, mainly focus on the residue feed intake. So this is actually our university bee farm. Right now, we are having about 700 beef cattle. And including 2 purebred, and then there is our university own crossbred. And since 1999, the University of Alberta, a group of researchers in bovine genomics and genetics have been genome typed all those cattle and aim to identify genetic markers for the residue feed intake. So the residue feed intake was selected because it's independent from the body weight and growth, but also it is moderately heritable. So you can see here that they actually moderated heritable during their different growth stage. Although there are DNA markers have been identified through past genome typing research, and the the genetic markers for this particular traits are still under testing. They need more population to clearly identify those gene markers that can be used for molecular breeding. Another interesting point is actually diet can affect R5 ranking. So if the animals are ranked high R5 doing growing diet, but when you change to the finishing diet, they might become a high R5 and vice versa. So at the time, it is unclear why those diet can impact R5 ranking. And that's where I started in 2005 to look at how potentially the animals, the rumen microbiota can respond to the diet and management, how that could be related to the feed efficiency, the R5 traits. So just give you some idea. So when we're talking about R5, so the R5 is actually evaluated by their estimated feed intake. The difference between their actually feed intake with their expected amount of feed when they reach certain body weight. So if animals eat more than their expected amount, that should be inefficient, usually is defined as high R5. If they eat less than their expected feed intake, it's called the low R5, so they are more efficient. So in the past, we actually have been looking at the very large cattle population and looking at how the rumen microbiota could be contribute to this feed efficiency traits. So I'm not showing, but in total, I actually have collected more than 2,800 rumen samples from 4 years. So, and then we have hundreds of samples have done looking at metagenome, metatranscriptome, and of course the 16S based compositional analysis. But in the meantime, we actually collect rumen epithelial tissue from 144 animals. We actually look at rumen tissue function because we think rumen as a whole, not just the microbiome, but also rumen tissue function play very important role. As also I mentioned before, we also looking at the tissue attached microbiota, whether they play a role in the tissue function. So in general, we still are trying to define the molecular mechanisms of the rumen microbiome, how they actually contribute to the R5, the traits, and whether we can potentially manipulate them to improve the feed efficiency. So I'm just going to introduce one of the most recent paper that we actually using the metatranscriptome-based approach identified how the active rumen microbiome could be associated with feed efficiency. So I didn't really introduce methodology, but it is we actually extract RNA and then we use the RNA sequencing to sequence all the microbial RNAs from the rumen microbiome. So what we found, and we found out actually there are certain microorganisms are identified in inefficient cattle. And what is interesting, we also identified there is more metabolic pathways and enzymes actually expressed in inefficient cattle. So what's the key message here? What we found, well, there is a methanogen identified as more abundant in the efficient cattle. That actually was a really interesting question. So this methanogen actually not only involved in the methane production, but this methanogen, there are species actually we identified, they were involved in nitrogen metabolism. So sometimes the methanogen that contribute is not just about they contribute to the methane, they actually also contribute to the nitrogen metabolism that actually can enhance the microbial crude protein synthesized in the rumen. But what's the key message here we want to say is actually the efficient cattle that actually has less diversity of the rumen microbiota and has less microbial activity. And what is interesting is I'll just go back that. I'll introduce, maybe talk about that later, because that's also found in other study. So what that this suggests that somehow similar, if you think about, we give cattle with antimicrobials, then one of the antimicrobial role is actually reducing the bacterial load, maybe reducing the bacterial diversity, maybe provide more substrate, because microbes and hosts compete for the substrate. So by looking at the less diversity, less the activity suggests that the cattle with more efficient, higher efficiency has a mechanisms to actually maintain less population in their rumen. But just based on our study, we cannot really say rumen microbes play a role, contribute to feed efficiency. So in the meantime, there are many other researchers starting to look at the rumen microbiota and the feed efficiency. So you can see here, these are the research published by the USDA group. They also looking at how different rumen proportion, the rumen microbiota associated with different fractions could be associated with feed efficiency in dairy cows, but also some information in here looking at with beef cattle. So they all identified respective rumen, the rumen microbial composition differ between high and low feed efficient, the cattle with high and low feed efficiency. So in the meantime, from UK and Irish, in the meantime, the Irish group also published a couple of the work looking at the rumen microbiota is indeed also associated with cattle feed efficiency. And one of the most recent UK work, this is just published last year, looking at also the microbial genes. So here is not microbial taxa. They actually look at microbial genes. They are associated with average daily gain, FCR, R5, and a daily feed intake. So you can see here, there's more and more evidence start to showing rumen microbiota play a very important role in, in the energy harvest efficiency in ruminants. And this is another study done by Israel group, Dr. Mezrach, and he has actually focused on dairy. We actually have done lots of work parallel. So he has published this work looking at the metagenome of the rumen microbiome, and what is very interesting, they find out, similar as our meta-transcriptome work, in the efficient dairy cows, they have low richness. So there's a less diversity. Although they have less diversity, but they seem like have the metabolites more likely contribute to the host growth. But you can see that in the inefficient cattle, they have more diverse richness, but their metabolites output are significant different from the efficient cattle, and those metabolites somehow do not directly contribute to the cattle growth and production efficiency. So you can see here, it suggests that those microbes and their metabolites could play a very important role contributing to the feed efficiency. So based on those evidence, the question is, does rumen microbiota play a role in feed efficiency of cattle? Yeah, I think yes, it's likely. So then if it's true, the question is, can we manipulate rumen microbiota or microbiome to improve feed efficiency? So at the time, the first idea we came out is, if we actually feed inefficient cattle with the rumen content from the efficient cattle, that will change feed efficiency because those microbes are efficient. So we did experiment a couple of years ago. We actually did a rumen swap study from 10 high R5 and a 10 low R5, because based on our hypothesis, if we switch our cattle, if we give the low R5 animal rumen to the high R5, they should change. well, that's our kind of a dream or goal, because I thought at that day, if I can find out that, I might be rich. I can sell all my rumen contents to improve feed efficiency, because rumen contents usually is a waste. Anyway, but that was actually a nightmare after the study. So in the experiment design, we had actually as a control, so we actually had exchange between low, 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 high, 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 low, high, high, low, and high, low, high, low. So actually we had 4 pairs, but then we have a 2 compares of control. So after this transformation or transplant, we got results is miserable, because their changing pattern is so different. And after that, their average daily gain measurement is also different. The VFA is also different. And the only conclusion we can draw is that each individual rumen actually reestablish very differently, depends on their endogenous, originally endogenous rumen microbiota, and also depends on their exogenous microbiota. And doing this experiment, we found actually there's a huge individual variation, and is very significant individual response to the exogenous rumen microbiota. This is not surprising. And when you think about future rumen manipulation, and you can see here, there is more and more evidence start to showing this huge individualized rumen microbiota. This is actually what we reported in 2009. We indicated there's a large variation among animals when they actually have the same diet, and they are just individually different. This is the Dr. Mizrash's study looking at composition, also look at the individualized rumen microbiota, but this is one of the most recent study when they're looking at more than 750 animals. They identified huge individual variation among the population. Most importantly for the rumen microbiota, each individual, as I said before, not about that pH, this is further confirmed that microbial composition shift very differently individually. So you can see here there's 4 cows. This is a research done by Netherlands. You can see that how individual cows microbial composition shift in response to the feeding. So this is also another study we did in the past. I had joined a PhD student with one of a top organization in China. We look at meat lactating cows, and we look at their rumen microbiome. And what we found out is the pan microbiome is also important to the traits. We actually identified both pan and the core. If you remember the 2 circles I drew, the core microbiome means all the cows have the same bacterial population. And then for the pan means only individual. They all contribute to the performance traits. So this has raised the question how we are going to manipulate them to improve that performance. So what are the mechanisms? So what are the factors may contribute to those animal-to-animal variation? So I'm talking about maybe pH, but what about rumen size? The second question is what about host genetics? And what about animal behavior? So for example, about their sorting, about their eating, and so on. So in the past now, we further deepen our research to look at what could be the genetic effect on the rumen microbiome. I'm not going to go through detail for this study, but we actually we used both metagenome and metatranscriptome, and we have identified that microbiome actually is breed specific. Here is Angus, Charolais, and hybrid. So when you look at, this is only showed composition, but when you look at the functional aspect, they also somehow segregated depends on the cattle breed. So host seems have a factor on the rumen microbiome, but through what mechanisms? So this is a most recent study we have just published, that we were able to identify the heritability of the rumen microbiota. So here you can see here we identified that 55 bacteria and 3 archaea, they are actually moderate heritable with using the calculation for additive heritability. And we also find out the members belong to Firmicutes phylum are more heritable, and members belong to phylum Bacteroides are more likely affected by the diet, okay? But what is more important, this is a table, I'm sorry if it's actually a little bit crowded. So here we actually identified some SNPs that associate with those heritable microorganisms, but they also associate with traits. Sorry, this is actually in the back. I cannot really see. So what we are doing right now, we actually are validate those SNPs in a larger population. They are associated with heritable microbes. And also, for example, here I can see DMI and FCR. So whether those SNPs actually can potentially be used as a genetic marker for cattle breeding and selection. So I actually look at this gene, the KCNK18. What is interesting, they actually only express in the rumen, not in the liver and muscle and the fat, based on our recent study, suggesting some here that some genes actually are rumen specific we can use for future molecular breeding process. So this is again another 2 studies I like to share with you that people also reported that host genetics can affect the animal performance, especially here in the methane. And this is the same study, very similar study, but they focus on methane. But in this paper, they also actually look at the feed efficiency traits. This is actually a paper from the ruminomics, and you can see here Dr. Mizrach has been involved significantly. So this is a joke, actually. We are not competing, but we actually, my student refused to send this to Science Advance because there was no impact factor. So, and I was joking with Dr. Mizrach that our paper was 2 weeks earlier before them, and then we just joke about this kind of a process, how we actually tackle the same research question, looking at the heritability or host effect on the rumen microbes. But are we there yet, right? So here are the concerns I actually have, is because the rumen microbiota we use is not quantitative traits. Many of us actually just use relative abundance and to do the analysis. So we really need to validate them over the multiple generation to determine the true heritability. We also need to define the interactions between those genetics and diet, but also we still need to know the mechanism, through what mechanisms. And also we need to think about, now go back to what I mentioned, rumen is very big. We only look at a small proportion of samples. We don't know the total microbial mass. We also do not know much about microbial enzymes and metabolites. Another thing is we actually lack of information on rumen size, passage rate, and nutrient absorption capacity in genetic variation, and how those all differently perform during the developmental stage, winning stage, growing and finishing, and so on. There are still so many unanswered questions, okay? I know that my time is limited, but I want to finish them in 2 minutes about some other efforts we made. So in the recently, we have been working on trying to identify how many rumen or how many gut at what extent microbiota contribute to host performance, and at what extent host contribute. So I'm going to introduce this 2 study. I'm not going to go through detail, but I'll just give you some ideas here, what we identified. Here you can see here that rumen microbiota actually only explain 70% or 18% of the milk protein yield variation we observe from the large population. And rumen microbiome, when you look at microbiome, that goes up to 22%. When you look at rumen metabolome, we actually did metabolize, they can actually explain up to 30% of the variation of the milk protein yield. But when you look at the serum metabolome, which is derived from host, they actually contribute about 27. So I believe they all actually work together to contribute to the host performance, similar as a genome biologist paper we published. We actually identified here that the host, actually the transcriptome and the regulator mechanisms that contribute almost 70% to the rumen development. And actually rumen microbiome only contribute up to 30% to drive that process. So here it's very important for us to think that microbiome is not just can explain everything. It's very important for us to think about the overall function, including the rumen movement, their physiologic healthy status, barrier, digestive, and absorption, but also think about the feed efficiency. Feed efficiency is not about, rumen is important for digestion, but also muscle growth, liver, all different functions can all contribute to protein turnover, their energy metabolism, heat resistance, and so on. They all contribute to the traits. So this is actually what we did. We expand our rumen work, look at different tissue, and we actually identified how different tissue could also contribute to the different feed efficiency traits. So what I want to say that actually the animal work as a whole systematically contribute to the traits, and microbiota is only just one of the factor. So I'm going to finish my slide with, in the future, that we need to consider when we look at the performance, we need to think about the cattle from the feed, how they affect rumen microbiota. But in the meantime, how those feed affect cattle physiology, behavior, their welfare, performance, health, they all contribute to the traits. Okay, so this is my last slide. I'll be there yet. I think that really depends on how we identify the drivers. So the passengers maybe just come and go. It's really the drivers are important. So are we able to identify the driver yet? I don't think we're not there yet. We still need more research to identify the biological meaningful function aspect of the microbiota, that they are active and their function contribute, they could be classified as driver. We then link them to different function of the host and the traits. And we really need to do more analysis, not just one population. We really need to combine data globally to identify whether the microbes are the driver, and to determine when is the critical window we can manipulate. And we actually, it's very difficult if we don't know when to manipulate for the production. Last, we really to determine the causal effect before we can actually manipulate rumen and gut microbiota. So thank you very much. Sorry for my long talk. Sometimes I do talk too much, and so I really appreciate and thank you for your attentions.
Thank you, Dr. Guan. It was a great talk. You gave a very good introduction about the rumen microbiome. And after your talk, I realized how challenging to manipulate rumen microbiome. So now we have some time for questions. Please, if you have any questions, feel free to ask.
Yeah, apologize, I couldn't talk about gut health, and maybe in the future I'm happy to do it again. You don't need to listen to me again, but
sure, yeah. Any questions?
Maybe I could jump in, Samat. That was a really enjoyable presentation. Thank you so much. I was struck by, when you were talking about the rumen and you were describing it as being this really huge area with a lot of different environments in that rumen. And when you talk about designing studies to examine the microbiome of the rumen, the way you design those experiments is really going to impact the kind of data that you get. And I'm interested in your field. Has there been a movement towards standardizing methodology, across research labs and approaches, beyond the peer review process? But is there a movement to identify best practices for these studies so that the data that's being produced is of high quality, and because I would imagine that that's going to be critical for identifying these kind of small effects that we're looking at identifying.
Actually, there were two EU-funded projects has been working on that. One of these rumenomics, they actually have done extensive standardization in terms of sample storage and sequencing and comparing different sampling methods on the rumen microbiota analysis. That was led by Dr. John Wallace. Right now, I think there's an ongoing EU-funded project led by Dr. Sharon Hughes at Belfast. They actually are also working on the standardized. So we all are as part of rumen what do you call rumen microbial genomics network, and that there has been tremendous efforts to do that. But it's very challenging, not just like a feces, right? Feces is so I'm not saying easy, you can be standardized very easily, but when you look at animals at different stage and when they have the diet are so different, and it's very challenging. When you compare the grazing cattle with dry lot can be also different. During our past experiment, when we tried to work on the growing and young animals, and when they were grazing, even use tubing is extremely challenging, and also we need to also consider potential animal welfare. So the only thing we need to think about is when we write a manuscript is really important. We should be aware what's the limitation that the data were generated and based. So we need to really describe that clearly, how much conclusive conclusion we can draw.
Dr. Guan, I'll ask a question. Larry Reynolds in Animal Science at NDSU. I think you said that the microbiome explained about 30% of differences in feed efficiency, or at least efficiency, however you were measuring that. And I just wanted to point out 30% is a huge number. If you could change efficiency by 30%, that's a huge number.
Yeah, apologize. It was not very clear. For that study is for milk protein yield. It's not a feed efficiency study. Apologize. And we actually have not been able to generate data. But if you read UK's paper, that they claim rumen microbiome contribute up to 80% of the variation. And I'm very conservative because the digestion in total itself only contribute maybe up to 10 to 20% of the feed efficiency. If you look at feed efficiency, so the digestion itself maybe up to 20, and usually we consider 10. So we were not able to define that yet. We actually now doing some modeling based on our very large data set trying to identify how much variation the rumen microbiome we see from the beef steer may contribute. I strongly doubt. So that's why you can see that I include more host analysis, because I believe for feed efficiency, the host will contribute, likely more significant.
Thank you.
So I have a question. Thank you very much for that presentation. I especially like the beginning and defining of vocabulary and adherence to strict guidelines to make reproducibility from lab to lab better. So my question is, I think, twofold. One is the observation that there was an individual aspect or a specific fingerprint of microbiota that you saw from cow to cow. My question there is, if the composition is different from cow to cow, which I believe is also consistent in the human and the mouse microbiota literature, does that necessarily translate to a different function? That's my first question. And my second question would be, are you interested, are you privy to how beef cattle researchers have investigated, and I think you showed some transcriptome data regarding expression profiles of the host, but specifically the rumen epithelium. Does it, since it's so close in proximity to the microbiota, is there an important dialogue between microbiota rumen and regarding the turnover? Is it as fast, like 5 to 7 days, which happens in human and mice? Is there a quick turnover, and is there this dynamic interplay between epithelial cells, for example, in the rumen? Those would be my two questions.
Thank you. Thank you. There are good questions. For the individualized microbiome, and it's pretty similar for all mice and human, and there's a lot of research. I think there's a very recent Nature paper just came out last week, talking about how individualized human microbiome respond to diet differently, and they associate with obesity, IBD. They actually did this huge kind of findings and how the individual microbiome drive that or lead to or associate with human health. So to answer your question, how different their function. So if you think about microbiota, there is a PNAS paper published in 2014. They look at human study. If you look at microbial composition, I think there is a varied about 60%, but when you look at metagenome, their difference is about maybe 20%, because many microorganisms share the same genes. But what is interesting, but when you look at metatranscriptome, when you look at their activity, the difference become about 40%. So what that mean? That means the composition difference may not necessarily lead to the functional difference. However, it depends on that individualized composition. I'm not for sure whether that makes sense. So if we only compare presence absence, they may not necessarily change the function, or high or low, they change the function. But it depends on the membership. If it's ABC, CBD may not be different, but if it's EFG, BCG, it may have difference. So it's very, very complicated, even based on the human research. For your second question about rumen turnover, I think the rumen turnover still largely driven by the host. There are a couple of researcher groups right now. Well, we don't have the capacity, but I'm aware that because rumen epithelium has these 4 layers, and that the top layer may not really necessarily directly respond to the mucosal or attached or epimural bacteria as what we usually see in the lower gut, because many people are interested in immune function of the rumen epithelia, and then the recent study shows that all the immune molecules likely in the third, what's the name now? I don't remember. Actually, I have a slide about rumen epithelium in the third and the basal layer. So I think that how those 4 layers has been differentiated, how the cell shape differ among those 4 layers are actually largely unknown. So one of the idea I had a couple of years ago is to see if we can do a cryo dissection to actually isolate single cells of 4 layers, and also look at different development stage to understand whether, you know, what is the regulatory mechanisms. But of course, I don't really have funding to do this because it's rather fundamental. But however, I do know some researchers are working on this aspect right now, and especially recently, very popular technology. People use single cell sequencing. They actually compared the different cell population in the rumen, but they haven't reached out yet to look at different cell population in different layer. So to answer, well, sorry for the long answer, but to answer your question, whether the microbes actually affect that different layer proliferation and the rumen and turnover, we don't know yet.
And vice versa, perhaps those layers of epithelia are talking to the microbiota, and there's this give and take dialogue, perhaps.
Definitely.
Okay, great, thank you.
So if you don't have any questions, if you have any questions, please, you can send it to me so I can forward to Dr. Guan. Now it's 1:35, so let's end our seminar here. And our next seminar is scheduled at December 9th, so please watch out for the announcement. Once again, thank you, Dr. Guan, for giving such a great talk and taking your time from your busy schedule to give us this talk. Thanks.
You're welcome. Thank you all. Well, if you have any suggestions, advice, and feel free to email me, and I'm happy to work with all of you.
Thanks.
Great. Thank you.